Hello. So, picking up where we left off last time, we're going into macromolecules and more specifically proteins and nucleic acids and the major points of both proteins and nucleic acids, how they relate to each other. We were talking a little bit about lipids, carbohydrates, fats, triglycerides, and those sort of things. And we'll get to that a little bit later on. To just to recap, basically, nucleic acids are the building blocks of proteins. You'll see individual nucleotides. There's only four of them with base pairs from adenine to thymidine and guanine to cytosine in a double-stranded DNA molecule that codes for a single strand RNA molecule that's processed further for a protein in the form of amino acids where three nucleotides will correspond to one amino acid. And then you have amino acids that will link up with peptide bonds to form a protein. And at that secondary structure, what you have is repeating units of amino acids. And you'll start to get into the tertiary, more complex structure where the protein will assume some function and structure, and that's the tertiary structure. Also, proteins can have a quaternary structure where you have multiple tertiary structures that interact with each other to allow for the function of that particular protein. So you can say the function of the protein, the structure of the protein, you can trace it back to the single DNA molecule. Absolutely. That's four nucleotides in some kind of repetitive unit. Those four nucleotides are processed into amino acids, three at a time, and those amino acids have specific properties that allow some type of function to arise when multiple amino acids come together in some type of way that allows for complex interactions. And you have alpha helices and beta sheets and other types of more traditional protein structure that will dictate the type of function, and that is contingent upon the nature of the amino acids themselves. So our proteins generally are stable in some sense, where there are so many different types of bonding patterns that keep the amino acids together. The peptide bond itself, a carboxyl bond bonding the amino terminus end of one amino acid to the carboxy terminal end of the other, will in itself is a strong bond. It's a form of a covalent bond that just links the amino acids together. Now you'll have stretches of those peptide bonds linking amino acids together, anywhere between 10 and 100, or could be a lot more, anywhere between 100 and 100,000, depending on the final product of the protein. And that has some type of inherent stability associated with it, with not only the covalent bonds, but also other hydrogen bonds, interactions with the proteins with water, the van der Waals interactions, which are weaker interactions, hydrogen bonding patterns that will are very weak interactions, but yeah, and also ionic interactions like salt bridges and disulfide interactions that are also ionic interactions. They're not very strong bonds like covalent bonds, like carbon-carbon bonds, because there's so many of them when they accumulate, especially in some specific domain of the protein. It allows for a localized um, domain of that protein, or the protein itself, to be very strong. So proteins have some type of function. That function is predetermined pretty much in the nucleotide sequence. Absolutely, when you have some sequence of DNA Say you have 3,000 base pairs of DNA in a row, um, and it's processed in the messenger RNA, where the messenger RNA, say it's 2,000 base pairs, because as you know, in nucleic acids, DNA, and even RNA, you have parts of the gene that don't code for a protein. They're called um, introns. That we know now has some type of actually somewhat important physiological function. The exons themselves are the ones that directly lead to the amino acids that will lead to the formation of the protein. Now, what you have from a 3,000 base pair DNA sequence to a maybe 1,500 to 2,000 base pair single-stranded messenger RNA, and you have further processing. Messenger RNA itself has a lot of processing. It's shuttled outside of the nucleus in the cytoplasm, interacting with other proteins, in particular the spliceosome complex, that, for one thing, takes out the introns and does other things as well through a complex interactions of many proteins. And then what you have is a processed messenger RNA that is ready to be translated into a protein. Now, this translation occurs when that single-stranded RNA will interact with a ribosome that is itself a protein unit, a huge complex that it will pretty much have a pocket for that messenger RNA, where it will one at a time read the three nucleotides and match a amino acid to that on one part of the um, ribosome. It's called transfer RNA. It looks like a clover leaf, where the bottom has complementary bases that is complement to the messenger RNA, and at the top you'll have the complementary amino acid that corresponds to the complementary DNA molecule. And then when you have that amino acid, that is then switched over to another part of the ribosome where that amino acid is left. It goes to a different part. There then opens up a place for the another amino acid to come in based on that messenger RNA sequence, where the ribosome is moving from a 5 prime to a 3 prime direction, or from the amino terminus to the carboxy terminus, and where you have a formation of a protein. You have stop codons that are in the messenger RNA will tell the ribosome to stop translating. You have start codons. For whatever reason, you have start codons that are or start codon or amino acids that are start codons. There's one called methionine that will signal the beginning of the protein. And that's very important because it tells the protein machinery where to start translating the protein. Also very important because if we want to study this, we know where that protein sequence starts, so we can backtrack and deduce its mRNA sequence and then its DNA sequence, so that we can understand the protein function or something about the protein on the nucleotide level. Now, this this process called transcription, where you're getting messenger RNA from the DNA molecule, and translation, where you're getting the protein molecule, the sequence of amino acids from the messenger RNA translation, is what we've been calling the molecular biology of DNA. It was basically worked out into completion in the mid to late 60s. Just a brief history. DNA wasn't discovered until 1952 by Watson and Crick, where they deduced the sequence of the double helix, which is a story. They won the Nobel Prize from it. They found out that the DNA is double stranded and that you could always depend on one nucleotide pairing with another one, and that there's a sugar phosphate backbone that stabilized the molecule, and the molecule itself, as a helix, was in the formation of a staircase. So that was sort of a big thing because around that time, years before, there were a lot of people that were trying to find out what DNA is. We knew that there was DNA. We knew that it was a nucleic acid. We knew it was a genetic material, but we structurally did not know what it looked like. Linus Pauling, who mostly studied proteins, was also looking at it too. He thought that maybe that DNA had three strands, and it seems like Watson and Crick sort of found out first, but they found out in kind of an unconventional way. It wasn't the structure that they deduced was based on an X-ray diffraction pattern from another scientist, Rosalind Franklin, who was a biochemist in England, and she basically showed a pattern of DNA that was very symmetric. And one of them, I think it was Watson, saw that image and immediately knew that DNA was double stranded and that there was a base pair rule that followed. There was some discussion about that sort of thing. Rosalind Franklin really never got credit for it. If it wasn't for her, perhaps they could have discovered it in another way. But it was based on that was sort of the hit the nail on the head, and where Watson and Crick decided that they could publish the paper, and the scientific community accepted it. And after that, there's a lot of molecular biology. In the 50s, we were still in its nascent form. Come the 60s, we were understanding how we get from DNA to RNA to protein. And around the 70s is when it really started taking off. And it wasn't really until the late 1980s or the early 1990s when we really started understanding this process, when we could experiment in the laboratory from the discovery of polymerase or polymerase chain reaction, where we could amplify the DNA and study it. It was discovered by Thomas Mullis, then at the University of California, Los Angeles, and he discovered it, won the Nobel Prize. It was probably one of the most important discoveries in this and in that 20th century. Ever since then, molecular biology has sort of taken off, and now we're kind of looking at more of the regulators and the players of the translation and transcription process to see how genes are regulated, how proteins are regulated post-translationally after they've been translated. Several mechanisms of regulation, especially as it relates to disease status. Just a second. So you have Chargaff's rule that basically says that G pairs with C and A pairs with T, and the GC content of a DNA is going to be higher than the AT content of DNA. But another point to make is the stability of DNA and messenger RNA itself. RNA itself, they're not as stable as the protein. In fact, DNA is more stable than RNA. RNA is single-stranded because what happens is after the double-stranded DNA molecule is produced after meiosis II in the cells in the nucleus, it harbors the information that's going to lead to eventually a protein. So it's very stable. The bonds between the nucleotides are hydrogen bonds, three hydrogen bonds between guanine and cytosine, two between adenine and thymidine, and there's other bonds as well. Those are actually fairly strong bonds. But what happens is, in order for a messenger RNA molecule to be made, that DNA has to be opened up in such a way where RNA polymerase 2 has access to those nucleotides, where it can read the complementary nucleotide and produce it in the form of messenger RNA. You have molecules like topoisomerases, helicases. Topoisomerases are protein molecules that unwind the DNA, they unzip, they cut the DNA and allow for access RNA polymerase, where helicase does something very similar as well. There's other mechanisms too, but those are the two big ones. So when you have an open DNA molecule, then you'll have this RNA polymerase passing along through from the 5 to the 3' end, and then attaching the complementary base, where it will produce a single-stranded messenger RNA molecule. And from that, you'll have a single-stranded RNA that's shuttled out of the nucleus by transfer ribosomes. This is called a transfer RNA that has a place that will bind that single-stranded DNA molecule that is it itself is basically a messenger RNA molecule, where RNA polymerase will read that, process out all the junk that it doesn't need, and produce a molecule, a single-stranded molecule that is not as stable. Now, the big difference, the biggest difference between RNA and DNA is the actual base pairing. You have the GC base pairing in DNA, and you have G and C and A in messenger RNA, but you don't have thymidine. So for that, you have uracil. Uracil replaces adenine for that, so you'll never see that. Now, that's going to be important when you're looking at the amino acid code that codes for that protein, or that amino acid that will eventually lead to that protein. So that's going to be it today. We have a few more minutes. Let's finish off with a nice diagram where I can give you the process of transcription and translation together. And we'll look at YouTube. All right, I'll just do transcription. Here's one right here, transcription. Okay, and then what we'll do here is I'm going to insert this. To my screen. I'll share my screen. We'll look at the process of transcription.
DNA transcription and translation.
He knows a lot about the science stuff. Professor Dave explains.
Now that we understand the structure of DNA, it's time to understand exactly how this molecule codes for a particular organism. How is it that a single cell containing a specific set of genetic material will result in the development of a fish or a cat or a human? To understand this phenomenon, we have to learn about transcription and translation. This is the collective process by which the genetic code is read by enzymes in order to produce all of the proteins in an organism. A chromosome is a very long molecule consisting of many millions of base pairs. Most of these bases don't do too much, but certain portions of the chromosome are special. They are called genes. These are the parts that code for different things. In a human, a gene will be on average around 10 to 50,000 base pairs long, though the longest is 2.5 million base pairs. And when a gene is expressed, a specific protein is produced. So how does this work? The first step is called transcription. This is the process by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA, or mRNA. To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific sequence within the gene, which is called the promoter, and pries the two strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from the gene and the DNA is returned to its original state. But we have produced an mRNA. This carries with it the information encoded in the gene. And after a few quick modifications during RNA processing, it will leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome. This is where translation occurs. During translation, the mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid. The arrangement of the nucleotides into these codons is called the reading frame. Since there are 4 bases and each codon has 3 letters, 4 cubed gives us 64 different possible codons, which is more than enough to code for all the amino acids we need. Here is a table of all the mRNA codons and the amino acids they code for. Notice that there is some redundancy, with multiple codons resulting in the same amino acid. But there is no ambiguity. Each codon corresponds to a particular amino acid. Notice also that some of these codons are special. AUG is the start codon, which initiates translation by coding for methionine. And these 3 are stop codons. These are the ones that terminate translation. Translation will occur inside a ribosome. The small ribosomal subunit binds to an mRNA and an initiator tRNA, which adheres to the start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. Then the tRNA that corresponds to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid, which becomes covalently bound to the methionine from the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifted over, making room for the next tRNA. The new amino acid links to the first 2, and this process continues all the way down the mRNA. As tRNAs enter and exit the ribosome in a sequence that is dictated by the codons on the mRNA, a polypeptide chain will grow. This continues until a stop codon is reached, at which point the completed polypeptide will swim away, most likely entering one of the cell organelles for folding and further modification. So in this 2-step process, DNA is transcribed into an...
Okay, that's about it. Thank you.